Thank you, Richard. Good afternoon, everyone. So yes, I have the privilege to lead a research group on the 20th floor of this ugly building next to the Shard that you can see when you come out. I always say we have half the view of the Shard for free. If you want to drop an email, I'd be very happy to show you around. Basically, we live in an image-rich world where data can become images and images can become data. This is easy to spot in the terms of probably there is a city around here somewhere. And basically, in our center, we use the power of image analysis to formulate hypotheses on biology. We use artificial microenvironment, stem cell biology, and image analysis. But there is one room in the center where we don't start from hypothesis, but we start from data. So in the few minutes I have, I would like to convince you that you can use image analysis to really characterize cell behavior. And we're building workflows and pipelines to automate this process. So you can imagine putting relevant metadata that correlate with the cells you are observing, using high content analysis to obtain features quantification of the cells, and then basically analyze this data. And this is where the community here is very good for us in partnering, because we can provide a lot of data and a lot of curated, curated data that can be analyzed with algorithms in AI. And there is a big problem in reproducibility, and so AI can help a lot also in the meaningfulness of analysis across sites. And imagine all of us here donating a piece of skin. The cells of our skin are reprogrammed to an early embryonic-like stage, and then they are characterized with imaging. So this is what we have been doing. We've been doing this together with other partners, and basically we use these type of solutions to quantify nuclei of the cells. You can also get information about how the cells proliferate, like this EDU marker, and their morphologies. Importantly, also context feature can be captured. So for example, you can distinguish a single cell versus a cell that is part of a party of 20 or so, and so you can get a sense of how cells like to be alone or be in communities. This is what's happening now at the Stem Cell Hotel. So we have a website I invite you to look up. We are a collaborative phenotyping space in which scientists are already coming to analyze their images, to use our devices, to discuss with us their experiments. So we're a very innovative structure in terms of what's available in academia generally. So we published in Nature in 2017 a paper within the HIPSI consortium together with the Sanger and the ABI and Dundee. And basically what we have done is taking 711 induced pluripotent stem cells, so these embryonic-like cells. And the question we were trying to address is precisely what I mentioned earlier. So how does a healthy volunteer cell look like? We see lots of papers where three cells from a patient's, three cell lines from three patients are described versus 3 cell lines from healthy volunteers. But really very little effort has gone into defining a baseline and the variance of the cells within a healthy population. And so this paper has delivered an appreciation of this variance across different type of arrays. Also a plot of the type of spots in the DNA that get erroneously replicated in the cell proliferation, and also a quantitative trait loci map. So we have a sense of where a mutation in the DNA can lead to a different expression of genes and proteins. And from our end, we've joined this project enthusiastically, bringing image analysis expertise. And so you can see that we tested, for example, different type of extrinsic environment on which the cells have been put. And this is a trick to sort of elicit more content. So again, if we come back to the analogy of all of us here, if I was asking you to stand, walk, and run, and then I would take pictures and movies, I would get enough information to sort of really capture how we differ to each other. So the question we're trying to ask is, how does the measures vary across replicates? How do they vary across cell lines from the same individual? And importantly, can we capture something unique of an individual that is reflected in their cells? And so we've done this on 110 cell lines from 75 healthy volunteers. And this gives you a bird's eye view of the sort of richness of this data, which is now published in Cell Reports earlier this year. We have a website curated by the ABI on hipsi.org where this data is available, and we can ask specific questions about how DNA, RNA, protein, and the images, even the raw data images, can sort of be collated and analyzed. And so this is the things we have done as an academic group. We have published papers on the image analysis segmentation pipeline, both in endpoint and in live images. And we have taken this together, bridging this with DNA and RNA. A big strength, as I mentioned, is the possibility of using both blurry dynamic movies that give you a lot of dynamic information but relatively poor content, combined with much higher endpoint information that can allow you to distinguish cell by cell and get characteristics of their behavior. And now we are pushing this further into not just single cell, but also how the cells are patterning together. So you can see how a group of cells confined in a 1 millimeter space on the dish and pushed with morphogens organized in a state that mirrors the early embryonic development with endomeso and ectoderm differentiation. And we're looking into ways to analyze this live in collaboration with Phase Focus. And I should say these are from Alice Vickers and Mukul Tewari in the center. In my group, we're also looking at 3D and analysis of 3D. So we're developing algorithm to sort of follow spheroids through their journey. Generally, spheroids are tested as an endpoint, but we see the value of actually observing the spheroid as it moves through. We have used a little gimmick that sort of subtracts 1 hour with the other to subtract the background. We're about to publish this. I'd be happy to give you some information if you're interested. But basically, the take-home message is we can characterize cells as single cell or in how they come together in 2D or 3D. And from this excellence in research, the hotel was born. We have been funded by the MRC and the Wellcome Trust in the HIPC project to get devices that are highly specialized, and they are offering the sort of top device available in the industry. So we also optimize the use of this device with the scientists. We have put in place a structure to recover costing. So this is a self-sustaining and offsetting the cost organization. But beside the aspect of properly more similar to a facility in academia, we are very passionate about building methods, building pipelines that can really solve questions about how cells differ one to another. And so we are bridging both methods of biology and software, but also we're bridging communities. We're bringing a lot of different expertise and interest, especially in the London startup arena, to King's and with a view really of working together towards translational projects. The promise of stem cells is in front of all of us. You can take a cell, you can mirror a state of disease, but again, how the cells differ from donor to donor is something that is really not enough on focus. And we would like to offer tools today to sort out this question. In the paper recently this year, we've managed to reduce the multidimensional space of these different features of how the cells look down to principal components, but also we've been able to extract synthetic features that can capture the extrinsic environment or that can capture an image of what's really unique for a specific donor. So you can compare the different type of environment, and you will have different values for one feature that is composed of a little bit of how the cell is round, a little bit of how it proliferates, a little bit of how it's shaped. So we managed to extract valid information that really pin down the environment. And if we ask for another feature, how the values differ from clone to clone from the same individual, we have a high correlation, which implies that we are capturing something unique about their intrinsic machinery. If you then correlate this across genes, you can understand that there is a relevant biological correlation, for example, to the transmembrane receptor of the cell for what concerns the environment. And if you look at single nucleotide variants of the DNA, you can predict different outliers' behavior. So what I propose to you today is to use very similar tools that capture intrinsic and extrinsic features, looking into difference in the age of the cells, distinguishing the senescence in terms of biological process in vitro with the actual aging process in the donors. And basically we offer a unique point to collect data of this sort in order to work together to their analysis. Thank you very much.